Katie Lawther. Yep, excellent. So Katie's in her final year of a PhD at Queen's University Belfast as part of the Huws lab group. Her key interest is in antimicrobial resistance in agriculture, and she explores this topic through a mixture of lab work and bioinformatics. So welcome.
Hi everyone. As mentioned, I am Katie Lawther, and my talk today will be on antimicrobial resistance associated with Northern Irish calf houses. So just a bit of background on my field. So I'm not going to go into detail on antimicrobial resistance, as I'm sure everyone knows quite a bit about it already, but just specifically within calves and dairy farms then. So dairy calves, disease is very high amongst them, and almost half of all UK dairy calves will experience diarrheal disease. Such enteric infections lead to high mortality and are the leading cause of death within calves on the Isle of Ireland. The antimicrobial resistance levels are particularly high in Northern Irish calves. So this table just shows the UK surveillance of resistance amongst E. coli isolates from calves and adult animals. And particularly within Northern Ireland, you can see that the resistance is high. For example, almost all isolates tested were resistant to neomycin. And this could be in partner with the high disease level, and also practices such as feeding waste milk, which contains antibiotics to calves. So the aims of this part of my study then is to contribute to AMR surveillance within NI dairy farms, to assess AMR levels in different areas of the calf house, and finally ask, do agricultural practices used by dairy farms impact on-farm AMR? So to do this then, we employed a multi-omic approach with culture-independent methods and culture-dependent methods. For this talk, I'll focus on the culture-dependent methods. So we started by farm sampling. So we visited 10 Northern Irish dairy farms across the 2019 calving season, and we visited them twice for sampling. On these visits, we collected a range of samples, including calf concentrate, which is feed, pen, bedding, swabs off the feed equipment, milk or milk replacer available to calf, and the calf feces themselves. So just included some pictures of the different variety of these things we've seen. So the pellets, and then the trough that's widely available to the calves versus some automatic feeders. And we also visited the farms a third time for surveys. So we accessed the farm's medical records. We did a survey of the calf house that included the setup of the house and what materials were used, how big it was. And we did a large survey on the farming practices. Just going into that last survey a bit more then, the farming management, we asked them a lot of questions and sampled a lot of this data, which is more than 90 parameters, including the size of the dairy herd and what mortality rates they have, how they manage calving or birthing, the setup of the calf pen itself, the calf health status, so vaccination status and health scores of the animals themselves, what feeding practices the farms use, and a lot of information was collected on the hygiene practices, so the frequency of cleaning, how they cleaned, and how clean the actual calf house was. So once we'd sampled then, we looked at testing for the AMR levels. We selected 7 antibiotics for this, which was amoxicillin, erythromycin, colistin, fluorophenocal, neomycin, tetracycline, and trimethoprim. The reason why these were selected, so 6 of them, excluding colistin, was used on farm and applied to the animals, and colistin was added due to its importance in human medicine. So to do this then, what we did was we took the samples, and for the milk concentrate and the different environmental samples, we plated them onto Muller-Hinton agar, which contained one of the 7 antibiotics. For colistin, we used McConkey agar. The fecal samples were processed using the same method, however, they were processed anaerobically versus aerobically for the environmental samples. Once the plating route was completed, we counted the colonies, assessed CFU per mil, and finally we picked some of the resistant isolates. These resistant isolates were then further characterized. So you can see the plate started with a lot of diversity. That's a fluorophenical plate and a neomycin plate. These isolates were then picked and streaked to purity. Following this, they underwent DNA extractions, PCR, and 16S sequencing. And then we did multi-drug resistant testing. So we tested the resistant isolate against all of the other antibiotics. And then finally, we gram stained them. So just to move on to results then, I'll quickly go over this is just a snapshot of the survey results. It's a lot of parameters, so for this I've just selected a couple of interest. And I want to highlight 2 farms that are examples of some extremities. So farm 2, for example, they had 100 animals calved that year, and they applied 36 applications of antibiotics. They cleaned their pens between 10 and 12 weeks, and whenever they were cleaning, they removed all material, washed the pen down, and used disinfectant. They're also frequently cleaned their feeding equipment. That was after every feed, and again used hot water and a chemical. And they uptook 7 out of the 7 vaccines available to them. In contrast, then, farm 4, they calved 175 calves. They only reported 2 uses of antibiotics. They were unsure how often they cleaned their pens, and they only removed the material. They didn't use any type of disinfectant. They thought they cleaned their equipment approximately monthly, and they weren't sure what method they used, and uptook 3 out of the 7 vaccines. So this is some of the AMR level results. What I have on the left axis is average AMR levels. What that means for this study is the CFU per mil of resistant organisms versus the total bacterial count, which was Müller-Hinton without antibiotic. So this is a busy graph, but what I want to really impress on you is the diversity between each of the farms, and the quite spread across all of the antibiotics. And the farms varied a lot, and some had very high resistance and some quite low. Looking at a similar graph, but this is by sample type as opposed to farm. And the pen, bedding, feed equipment, and milk all show low resistance, and they were quite similar. And that was in great contrast to the concentrate and the feces. So they both had high resistance levels. The concentrate showed a wide variety again across the different antibiotics in comparison to the feces that was dominated by neomycin, tetracycline, and trimethoprim resistance. Looking at the multi-drug resistant results then, this was of 216 isolates that were tested against the 6 antibiotics. Colston results aren't included in this, as that is what I'm currently working on. So this is the number of antibiotics that each isolate was resistant to, so 1 through to 6. And the worrying thing from this results, I guess, is that the high number of isolates that were multi-drug resistant, particularly the number of isolates that were resistant to 6 of the 6 antibiotics tested. Looking then at the different combinations that we observed of multi-drug resistance, the most common was neomycin and trimethoprim, closely followed by the multi-drug resistant pattern against 5 antibiotics of amoxicillin, fluorophenical, neomycin, tetracycline, and trimethoprim. And looking at the Colston results I do have then, so this is the samples that did contain Colston resistance, and that was 5 out of the farms tested. So what we have is farm number, the sample type, the Colston resistant CFU per mil or per gram, and again the AMR level as on the previous graphs. What I would say is that sometimes the AMR level was high. So for the first line, for example, it was 100% of the isolates were resistant to Colston. However, the CFU per mil was actually quite low. So it's important to, whenever I'm doing the analysis, to take both things into account. And what I've highlighted in green, slightly different then, is the feeding equipment and the concentrate feeds. So as you can see, most of the samples that contain Colston resistant were of this type, and three feces. So the key points I really want to highlight from this talk then were the farms across Northern Ireland showed varying AMR levels. However, all farms contained resistant organisms to 6 out of the 7 antibiotics tested. Going further, that then half of the farms tested contained colostrum resistance, with concentrate and feed equipment being the most common area for identification. So we are suggesting looking further into that there may be a link between these areas and colostrum resistance. Over half of all aerobic isolates, which is 66 isolates, were multi-drug resistant, and even higher than over 93% of the anaerobic isolates were multi-drug resistant. The further work then, what I'm coming towards the end of my PhD, and the main thing is starting on the stats, so trying to make correlations between the AMR profiles that I've described here and the farm management styles, and analyze the metagenomic sequences. So all of the samples that I've described today have underwent metagenomic sequencing analysis, and finally look at the 16S data to try and look at the taxonomy of some of these isolates that I've described. So I just want to finish by thanking the network for allowing me to present today at this conference. I would like to thank my funders, and this work was completed in collaboration with AFBI and the Optihouse project, and my supervisor Sharon Huws and the fantastic Huws lab that I work with. And thank you very much.